I will start in like 2, 3 minutes. Right, so I'll share my slides. Okay, so this is the 10th live session of this course, NMR spectroscopy for structural biology. And in this session, we will look more into how the drug discovery process goes on and how NMR helps in this drug discovery process. Just 1 second. Yeah, okay. So first, the drug discovery process takes many years and involves many failures and a lot of uncertainty. Most often, the development of a new medicine starts when basic scientists learn of a biological target, that is, a receptor, enzyme, protein, or gene. So, basically, at the initial stage of the development of a new medicine, they find out that what is the gene that is causing that defect, or is there a certain protein interaction that is resulting in the disease. So, once they choose the disease, they find a drug target. Suppose there is a gene that is defective, and when the transcription factor binds and reads that gene, it results in the disease. So at that time, the target for your drug becomes that gene or the protein interaction that is resulting in the disease. So that becomes your drug target. Then you identify a bioassay, which is a test used to determine biological activity. So, suppose you're going to have a list of possible drug molecules. You need to test how effective these drug molecules are in inhibiting the protein protein interaction or reading of the gene, or basically how effective this drug target is in preventing or reducing the effects of the disease. And that test is called the bioassay. Then you identify the pharmacophore. which is basically the structural features directly responsible for activity. So you identify the basic structural feature that your possible drug molecules need to have in order to be biologically active and interact with the gene or protein that you have identified as your drug target. Then then you will get a list of molecules which show low level binding. Then slowly the structure is optimized to improve interactions with target and show higher binding and selectivity and so on. Then you get a lead compound. And if the lead compound is not known, the structure of the lead compound can be determined from what is already known in nature. So the lead compound is a molecule that has some activity against the chosen target, but is not yet good enough to be the drug itself. So basically, after you identify the pharmacophore, that is the structural features that are responsible for binding, using the structural features of the pharmacophore, a lead compound can be designed. The lead compound will show some level of activity against the chosen drug target, but it is not yet good enough to be the drug. That is why it's called the lead compound, because it is a starting point. From the lead compound, the structure can be optimized more and more in order to improve interactions with the drug target. So basically, this entire process is kind of like fishing. So you can have many different possible drug targets, and you have a molecule that is basically suppose known to be biologically active. So you can kind of fish that molecule against the available possible drug targets to see with which it is showing some kind of low level activity. That will help you identify the initial drug target and the initial lead molecule. And then eventually the structure of the lead molecule can be optimized in order to improve binding and get closer to a better and better drug. This entire process takes many years. Better medicines are iterative improvements on current medications. So basically, you might have, so let's start at the process. So you start and then you have a hypothesis. Like, suppose there is a certain pharmacophore that you have identified that will be a good binding molecule. You design a molecule and you synthesis it. But in order for synthesis, you have to find out that what will be the synthetic protocol. The deduction of the synthetic protocol consists of many methods. It might be chemical intuition, which is your basic knowledge of chemistry. Then it can be combinatorial approaches, like you have a certain, suppose you have a certain structural feature A, which leads to binding, and also a structural feature B that leads to binding. Just 1 second. Right. So as I was discussing that in the process of drug discovery, 1 second, there is. Right. So as I was discussing, in the process of drug discovery, you basically start from a hypothesis. That is, you think that there are certain structural features that your drug must have in order to bind properly to the target. Then you design a molecule which can be a possible lead compound. And for designing, you use different methods. One of that is chemical intuition, which is the basic knowledge of synthetic organic chemistry. Then there can be combinatorial approaches. What this means is that if suppose you have a certain structural component A which causes binding and a structural component B which causes binding, you can join the two using some kind of linker and you have a molecule which will show better binding than A or B individually. So that is kind of like a combinatorial approach. Then you can have molecular modeling. So there are many softwares available now which are basically docking softwares where you can design a model which can bind perfectly into the receptor site of the protein. Or there are de novo methods, that is looking at the natural protein-protein interactions. You can design a drug molecule from scratch. So using all these methods, once the drug molecule is designed, it can be synthesized and then it is tested. So using bioassay, which is a technique for testing the biological activity of the possible drug molecule, uh, the testing is done. So suppose you have a molecule which shows binding, but not to the extent to which it can become a drug. Then you repeat the process. How do you repeat the process? You come up with a better design using different kinds of modeling. So this time you are seeing that a certain molecule is showing a low level binding. Then again, using your knowledge of chemistry, you can design a better molecule which will show higher binding. There are other ways of modeling and even using AI, which is quite common now, by which after the first set of tests, you can design the next set of molecules, which is a possible drug molecule. So every time you design a molecule, you learn from the bioassay or binding properties of that molecule in order to improve the binding of the current molecule. So like this, by iteratively repeating this process, better and better medicines are developed, which are improvements on the current medications. When we say better, that means there can be benefits like the potency, that is how much it can reduce a disease or its symptoms, the safety, that is the toxicity in our body, the tolerability, that is how much we are able to tolerate the side effects, or the convenience, that is something which can be orally ingested is much more convenient than something which needs to be injected into the bloodstream. So these are all qualities that can be improved in drugs. Historically, drugs were primarily discovered by empirical methods investigating substance or preparations of materials such as plant parts or plant extracts found in the local environment. So as we know, before all these softwares and docking mechanisms and modelings were available, people learned from nature, and the initial medicines were all based on extracts of plants or animals. These extracts were isolated and purified in order to find out that which is the active compound that is causing. Many cases, like in homeopathy and so on, plants are used as medicines. So what is the active compound in the plant that enables it to be used as a medicine? So many compounds like morphine, quinine, and so on were actually discovered after looking at these plant extracts. And these were known as naturally occurring classical therapeutic agents. Now, what we know that from these classical therapeutic agents, so you have the quinine, which was discovered from nature, and these biologically active natural products were structurally modified in order to optimize their pharmacology and drug properties. So quinine was structurally modified into chloroquine, where you have a chlorine group, and then also this six-membered ring was kind of opened up, and there was a further modification called hydroxychloroquine, where in the chloroquine at the end of this moiety, a OH group was added. So like this, from the natural molecule, more and more modifications were done in order to find out a better binding molecule. So now the new malaria drugs, they don't contain quinine, but instead they contain hydroxychloroquine, because that has been found to have better biological activity against malaria. So this kind of modification was done based on the understanding of organic chemistry or even from docking softwares. Now, one thing to remember is that the activity of a drug can vary very much depending on the exact configuration. So here you have two molecules, thalidomide, which are exactly the same except the configuration at this carbon. In this one, it is R configuration, and on the one on the right, it is S configuration. Now, the R configuration is the one that is used as a mild sleeping agent and also to treat morning sickness in pregnant women. But if by mistake the configuration at this site from R it becomes S, it becomes like a poison and causes a lot of fetal abnormalities. So just by changing the configuration at one carbon center, what is a medicine becomes something that is a poison. So the that is very important in drug discovery, is not only to maintain the molecular structure but also the molecular geometry and configuration. Now, there are NMR based strategies that can identify weak binding compounds, and using bioassay, these weak binding compounds can be developed into more and more potent drug like inhibitors and help us to find lead compounds in drug discovery. So, we have already seen in the last class how you can have a ligand and a protein. Now, if you look at the NMR of a ligand, the ligand is a small molecule, so its relaxation time will be very slow. And because of that, the ligand peaks will be sharp. Protein, on the other hand, is a large molecule and will have fast relaxation, so it will have broad peaks. The ligand protein complex is also a large molecule and it will behave like the protein, and so it will give rise to broad peaks. Similarly, in case of other NMR observables also, the ligand being a small molecule, it will have fast diffusion, and because the number of atoms are small in the ligand, the spin diffusion is also negligible. And in case of NOE, it will show positive NOEs. And protein and ligand protein complex will behave in a similar manner, which is completely opposite to the way in which the ligand behaves. In that, the protein and the ligand protein complex, they are both large molecules, so they will have slow diffusion and they will have negative NOE. And because there are many spins in both these, the protein and the ligand protein complex, the spin diffusion is also very fast. So basically, the ligand behaves in one way, and the protein or the ligand protein complex both behave in a completely different way in NMR experiments. So you can find out whether a ligand is free or if the ligand is bound to the protein using these observables like the relaxation time, the NOE sign, and so on. NMR can provide structure of a macromolecule at atomic resolution. This we have already seen that how NMR can be used to find the structure of a molecule. It also gives you an idea about molecular and supramolecular dynamics. So we have seen before in exchange type of NMR, when there is slow exchange, the peaks are well separated, and when there is fast exchange, the peaks kind of merge. So it gives you an idea of the dynamics of the compound as well. It can characterize weak interaction between molecules. We have seen this in the last class through experiments like STD-NMR, STDD-NMR, transfer NOE experiments, water log Z, and so on. We have seen that the binding constants can be characterized in the protein ligand complex. So we can find out about protein ligand interaction and also protein protein interactions. And the advantage of NMR is even for proteins that are embedded inside membranes. So you can see this is the GPCR protein and it is embedded inside the membrane. If you take it out of the membrane environment, the protein will lose its characteristic and it is not able to be completely purified from the membrane. We have seen in the last class that in order to study these membrane bound proteins, we have techniques like STDD NMR. So NMR can even give you ideas about this kind of membrane bound systems. So one of the techniques by which this binding of protein and ligand can be studied through NMR is chemical shift mapping. So protein ligand, protein nucleic acid, or protein protein interactions are the ones which we are interested in studying, because these are the interactions that lead to different diseases and if we can cause the drug molecule to bind with the target protein or nucleic acid, we might be able to prevent these diseases. So NMR can characterize these interactions from KD values of millimolar to nanomolar, as we have seen in the previous class. In some cases where we are looking at the protein, we might need to label the protein with 15N or 13C nuclei. So when we so suppose we are studying protein-protein interactions, we have one protein SKP1, which is labeled with 15N, in this case 15N, because we are looking at the 15N 1H spectra. SKP1 is binding to another protein SKP2. The binding sites are shown in red here. And we are looking at the HSQC spectra 15N versus 1H. When SKP1 is binding to SKP2, the chemical environment of these binding sites are altered. So if we look at the HSQC peaks, we can see that they are shifting. One of the peaks of this amino acid, leucine 65, is shifting, but leucine 44 is not shifting. That means leucine 44 is not a part of the binding site because it is not shifting as we are adding more and more of the SKP2 protein. But on the other hand, leucine 65 is part of the binding site because as we are adding more and more of this SKP2 protein, the chemical shift of leucine 65 is changing because the binding is causing a change in the chemical environment in the binding site. So observing these changes in 15N 1H spectra, which is shown here, or in some cases 13H 1H HSQC, 13C 1H HSQC spectra, we can find out the binding constants, the degree of binding, and so on of protein-protein or protein-ligand interactions. So this is typically done for small proteins having 100 to 200 amino acids. You can see here a 1H NMR spectra where the red spectra is in the absence of a ligand and in the presence of a ligand it is the blue spectra. So you can see there are some places where there are clear shifts. So in this case, in the absence of a ligand, there is a single peak, and in the presence of a ligand, two peaks are appearing. And also there is a chemical shift in this direction. So the nature of the spectra, the coupling constants, chemical shifts, everything is, and the intensity also reduces. Intensity is also changing. Similarly, if you look at the tryptophan side chain hydrogens, that is undergoing a shift in the opposite direction. So here the shift is in this direction. Here the shift was in this direction. So the direction of chemical shift can vary, but the thing is it will be different in the presence and absence of a ligand. Here also you can see for this, here the 1D spectra is shown, here the 2D spectra is shown, where the red peaks are the ones in the absence of the ligand and the blue peaks are in presence of a ligand. So you can see that there are some peaks which shift a lot. You can see this red peak is shifting such a lot. So this is definitely in the binding site because its chemical environment is changing so much. Then similarly, this peak is also, the delta delta of this peak is large, so it is part of the binding site. But if you look at this peak, the red and the blue are superimposed. So it is not affected by binding of the ligand, so it is not part of the binding site. So this will help you determine that which amino acids are part of the binding site in the protein. Now here, so we were talking about small proteins of 100 to 200 amino acids. You can have large proteins like dihydropicolinate reductase, DHPR, which is a 120 kilodalton protein. Now in this protein, what is basically done is that out of every 20 amino acids, 6 have a methyl group. And we can exploit the methyl group as probes. So you can see here the circles that are shown are the methyl groups that are in the protein. And they are distributed all over the protein and are part of the side chain. So, if in such a large protein, we are not able to study all the amino acids, but if we just look at the amino acids having a methyl group in the side chain, we will have a compact idea of the tertiary structure of the protein. So the 6 amino acids that have a methyl group in the side chain are some like alanine, leucine, isoleucine, so on. Uh, these can be exploited as probes. So instead of labeling all the 15N or 13C, we exclusively label the methyl group with 13. So methyl is CH3, that C is made into 13C. So there are ways of doing this by selectively labeling some compounds and feeding them to bacteria, and making use of the metabolic pathway of the bacteria to exclusively label only the methyl groups of the amino acids. So here you can see that if you had the HSQC spectra of all the amino acids, it would have been a complete mess. All the peaks would have been overlapped and nothing you will have been able to resolve. But if you look only at the methyl groups, it is a much more well-resolved spectra. There are different kinds of methyl groups shown in red, dark blue, and light blue here. And the different kinds of methyl groups, one might belong to alanine, one might belong to leucine, one might belong to methionine. So they will have chemical shifts in different regions. And that will help you further in finding out and resolving that which methyl group belongs to which amino acid and in what environment this amino acid is, whether it is in a beta sheet structure or it is in an alpha helical structure. So basically what this method of selectively labeling the methyl groups is doing is that it is simplifying a much more complicated spectra of the large protein. So this is one of the techniques of solving the NMR spectra of a large protein. Now we will look at some NMR-based drug discovery strategies. First of that is SAR, that is structure-activity relationship. So the SAR by NMR is based on the use of protein chemical shift changes to screen for low affinity ligands and the collection of structural information to direct a linked fragment approach to enhancing binding affinities. So what does this mean? We'll go by it one by one. So first we might have say 100 molecules which are possible low affinity ligands. We will screen the 100 possible drugs with the protein, and we have already seen in the previous class that STT-NMR is a technique that will give you the peak only of the binding molecules. So doing that, so suppose we have a protein with 2 binding sites. We are screening different drugs against the protein. Suppose this is drug 1, this is drug 2. And we are finding drug 2 is binding with KD of 17 millimolar. Then we are screening the second binding site of the protein. So suppose this is drug 1, this is drug 2. Drug 2 is showing good enough binding of millimolar affinities. This is your drug 2. Then against the second binding sites, you are screening drugs 3 and drugs 4. Since drug 4 is fitting better, it is also, this is drug 4, it is showing KD values of 20 micromolar. So for binding site 1, you have found a molecule having KD of 17 millimolar. For binding site 2, you are finding a second molecule having KD of 20 micromolar. So you have found 2 low affinity ligands for the 2 binding sites. Once this is done, the 2 ligands can be linked. So you can see this is the ligand molecule 2, this is the ligand molecule 4. You can link these 2 ligands. You can see here they are linked to find a third molecule which is a better binding of 15 nanomolar. So ligand molecule 1 showed bindings of millimolar. The ligand molecule 4 showed bindings of micromolar. But combining the 2 ligand molecules into one molecule is showing much greater binding of 15 nanomolar. So that is what the second part is, that the collection, you have a collection of structural information about what kind of ligand will bind to site 1, what kind of ligand will bind to site 2. Then a linked fragment approach is done to enhance binding affinities. So basically, you are linking the two ligands to form one molecule, which shows higher binding than any of the two molecules are doing individually. It is showing a binding of 15 nanomolar, uh, which is a very good binding constant. So NMR technique has been done for screening weak ligands. You have screened ligands which are weakly binding to each of the sites, and then structurally directing the chemical linkage of two low affinity ligands. So a linker is determined which will link the two low affinity ligands to create a high affinity ligand. So this technique is called SAR by NMR, that is structure activity relationship by NMR. Because you are using NMR to do the initial screening of ligands, which will be appropriate for site 1 and site 2. Now, in this strategy that we have discussed, there are some other way, subparts of this strategy, one of which is called as shapes screening. In shape screening, what is done is we assess the binding of a fairly small but diverse library of low molecular weight scaffolds to a drug target using NMR techniques that are based on observing the ligand. So when we talk about ligand based NMR techniques, we talk about, so we are not labeling the protein and we are not labeling the ligand also. We are observing the ligand to find out whether it is binding to the protein or not. So some examples of the ligand based NMR techniques that we studied in the previous class are the STD NMR, the water log Z, the NOE transfer, and so on. These techniques are used to screen a small library of different possible drug molecules, and the library that we are screening contains different kind of scaffold. So let's give an example. Suppose one we have in which two cyclic molecules are connected. A second kind is where three are connected. A third kind is where fourth are connected. So we are screening a lot of different molecules. So these molecules are different in their basic structure. So these are different scaffolds in order to find out which kind of molecule will have the best binding. These scaffolds are mainly derived from shapes or frameworks that are commonly found in known drugs. So using the knowledge that we already have from known drugs, the initial library of molecules are created. Just one second. Then we find out the initial weak binding. So there will be some ligands which will show initial weak binding, like suppose this one, which has a KD of 2 millimolar. Then using the hits, that means the molecules which have successfully shown weak binding, that is used as a starting platform for virtual screening of different analogs of the molecules. So we have found this molecule is showing 2 millimolar. Now, if we design a molecule which adds one extra benzene ring to this position of the molecule, and we screen that library, so we are adding the benzene ring first to this position, then to this position, and we are getting a second set of molecules where there are three cyclic molecules attached. We see that the binding improves from millimolar to micromolar. So after the initial weak binding compounds, chemical modification is leading us to another set of compounds which are better binding. Then we again try to improve the binding by adding another benzene ring to this site of the molecule or to this site of the molecule, and we replace sulfur by nitrogen. So different kinds of modifications are done in this way, and we get a third set of molecule which is from micromolar, it is improving to nanomolar. So basically, on the basis of the initial weak binding hits, a library of analogs with further derivatization of the initial core structure was screened to obtain higher affinity ligands. So you're just deriving and forming derivatives of the initial hit molecule to get better and better uh, binding molecules. Further substitutions of the hits results in compounds that are very potent inhibitors. 1D NMR screening was used to identify these higher affinity analogs of the weak binding hits. So this entire strategy is known as the shape screening strategy. Then you have something called NMR solve, where solve basically stands for structurally oriented library valency engineering. So what this means is that it is a design strategy that provides information for the construction of focused library against multiple related target. So in case of shape screening, we were randomly screening a lot of ligands. So in order to find the initial weak binding ligand also, we were randomly screening a lot of ligands based on known drugs. Then we were again derivatizing the initial hit and again randomly screening and so on. But the NMR solve strategy is not random screening. In this strategy, what is done? The binding site of the protein, the structure of the binding site is found out. How this is done is basically you have, uh, uh, so uh, when we say a protein family, we mean that one of the binding sites is the same for all members of a protein family. You can see the first binding site, which consists of 1, 2, 3, 4, 5, 5 edges. It is the same for all members of the family. And the second binding site is varying. So these kind of proteins which have one common binding site and the adjacent binding site is different, it's called a protein family. NMR solve exploits this property of large family of proteins that have adjacent binding sites of which one is conserved throughout the family. So if you manage to find a good drug for the one site, then it will act as a ligand for all members in that protein family. So basically, there are certain probes. So as we talked about, we can selectively label the methyl groups of the protein. Similarly, here we can selectively label some amino acids which are surrounding the binding site that is common in all members of the protein. So here you can see 3 amino acids which are radio labeled around the binding site. This is done in order to find out that what is the orientation of the binding site, and so what molecules will bind to the binding sites. There is a fourth probe shown in red here, which basically helps you understand that what is the orientation between two binding sites. So if you can find out that what is the amino acid structures between the two binding sites, you can construct a linker which will link the first binding site to the second binding site. So it will help you. So like we talked about SAR by NMR, where two weak binding ligands were joined by an appropriate linker to form a stronger binding ligand. So like that, you can construct the linker based on the information you get from NMR of this probe. So this probe will give you an idea that what is an appropriate linker which will join the ligands binding to each of these two sites. So you can see here the orientation of a common ligand mimic in the binding site of the protein is determined to provide the basis for the design of this linker, which is directed into the specificity site. So specificity site means the site that is different in the different members of the protein family. So in order to design a linker, this probe that we are selectively labeling here will give you information of what should be the structure of this linker. So NMR solve, this technique does not depend on random screening of a library of ligands. In this technique, before we decide on a library of ligands, the structure of the protein binding site is determined so that we can choose those ligands which will appropriately fit into that binding site. So here, before screening, we are doing a study of the protein binding site itself. So in this case, what happens? The NMR data for protein 1, so we have added probes in the binding site that is common in all members of this protein family, and it will help us design ligands which will bind properly to this binding site. So this NMR data of protein 1 can be used to construct possible ligands for all members of the protein family, because the ligand in the first binding site will be common in all members, and then by this red ligand, this red probe that you can see, this will give an information as to what should be an appropriate linker that should be um, synthesized in order to connect two ligands to give a better binding. So once you have the information from one protein, you can use it to have to construct better and better ligands for all the proteins in the same family. So this kind of thing is also called as fragment based drug discovery, or FBDD. So we talked about SAR by NMR, where we first find out a ligand which binds to binding site 1. We can find out through 1H15N HSQC. So like we said that in 1H15N HSQC, the peaks will shift if that particular amino acid is binding. So using the data from the perturbation of chemical shifts, we can find out F1, which is binding to site 1. Then we can find out F2, which is binding to site 2. So we have two fragments which are binding to two different sites, and we found the fragments by NMR based screening. Then we can find out the tertiary structure of the complex with the two optimized fragments. This tertiary structure can be found out by taking the NMR of the protein ligand complex. So we can find out that once the ligand is binding to the protein, what is the tertiary structure of the protein ligand complex. Then we can optimize a linker. We can do this by adding a probe in the linking region, which will help us find out what will be the best linker. And by linking these two fragments, we now have a bidentate ligand, meaning a ligand that is binding to two sites of the protein. And this bidentate ligand will have better binding affinity than either of the ligands are showing separately as fragments. So here some examples of bidentate ligands are shown, and this kind of method of drug discovery is called fragment based drug discovery, because you initially find the fragments and then the fragments are linked by an optimized linker. In order to find out what this linker should be, one strategy we have already discussed is radiolabeling the amino acids in the section between the two sites, so that based on the orientation of these amino acids, a linker can be designed. Another strategy is using ligand-ligand NOEs or intra-ligand NOEs known as ILOEs. So suppose you have a hydrogen on fragment 1 and a hydrogen on fragment 2. The NOE between these two hydrogens will give, which are known as ILOEs. You can see them here. So in this case, NOEs, you can see that there are, so you have labeled the hydrogens on fragment 1 as AB and so on, and similarly for fragment 2. So if you can see that, which suppose in this case 2A and 2B. So we have here 1B interacting with 1A. So that is within the same molecule. That does not help you. But suppose you have intraligand NOE, which is not shown clearly here, but suppose you have C, 1C interacting with 2D. Suppose you have a peak here. So 1C, this is interacting with 2D and E. So a ligand has to be, so the linker has to be designed in such a way that this side of the fragment 2 is facing fragment 1 and it is joining the D and E with C. So if we design the linker in such a way that the ILOE is observed, then it will be a better bidentate ligand. So the ILOE will help in constructing the best linker between the two fragments. Now, that is all about the basic process of drug discovery. But when this process is done, what are the basic properties that a drug should have? That is the pharmacokinetics of a drug. Firstly, a drug should be able to be absorbed properly. After absorption, it should be able to be distributed properly in the body so that it reaches the place where it needs to go and act. Then the drug should be able to be metabolized, because only when it is metabolized that it can have its activity. And finally, the side products of the drug must be able to be excreted from the body properly. So this is known as ADME, that is absorption, distribution, metabolism, excretion. The fifth point is toxicology studies must be done in animals and humans. So the drug cannot be toxic to us, only then it will be able to be a good drug. Now, physicians prescribe drugs on the basis of the characteristics of the medication and the probability that reliable and reproducible clinical effects will result. But the problem is that the drug response in different patients is different, and this leads to challenges in optimizing the dosage for an individual patient. Another problem is that most of the drugs are only effective in 25 to 60% of patients. So these are the two major problems. One is that most of the drugs are not efficient in all patients, and even if they are effective, the amount of drug needed for different patients, that is the optimum dosage, is different. So we will look more into why that is. Firstly, NMR has been used for the study of drug metabolism in vitro and in vivo. So NMR spectroscopy is very unique that it permits studies both in vitro and in vivo. By in vitro, we mean using body fluids like urine or blood or tissue extract or biopsies. And in vivo, we mean that when the human or animal is still alive, in like something like MRI. We know that in MRI machine, we are just put into the machine and the machine can scan us. So without taking anything out of the body, studying the metabolic products inside the body is known as in vivo NMR. So NMR spectroscopy is very unique that it allows both in vitro and in vivo study. Basically, there are certain proteins, enzymes called cytochrome P40 that are important for the biosynthesis and degradation of different compounds. So expression of a protein, expression of this cytochrome P40 protein is different in different persons. And the cytochrome P40 is what is responsible for the metabolism of drugs. So if this protein is expressed to different extents in different persons, then the metabolism in the drug in different persons will also be different. And this is what is resulting in a difference in availability and efficiency of the drug from person to person. So because of the difference in expression of the cytochrome P40, certain drugs are not, they don't have any effect on certain patients. And certain drugs, even if they have effect, the amount of dosage is different because the expression of this cytochrome P40 protein, which is causing the metabolism of the drug, is different. So there can be different sites of this metabolism. One of the major site is the liver, but the enterocyte, which basically are cells in the epithelium of the small intestine, is also an important site where cytochrome P40 is causing the metabolism of the drug. So we will look at the drug metabolism after the oral administration of a drug. So suppose you have, this is a drug called felodipine, which is used to treat high blood pressure. It basically relaxes the blood vessels so that the heart does not have to pump so hard and the high blood pressure reduces. Now, suppose the initial dosage of this drug is 100%. In the stomach, it continues to be 100%, but when it reaches the intestine, the enterocytes in the intestine contain cytochromes, and that results in that causes metabolism of the drug. So the availability of the drug in the intestine can vary from 45% to 15% to 30%. So due to different degrees of metabolism in different regions of the intestine, the bioavailability of the drug also varies. And in the liver also, we saw in the last slide that there is metabolism of the drug due to presence of the cytochrome P450. So in the liver also, the bioavailability decreases to 45% or 15% in different places. So this bioavailability is the one that determines the dosage. So as metabolism is happening, the drug is getting decomposed. So suppose in one place only 15% of the drug is available. So then we have to increase the dosage in such a way that this 15% is enough to treat the disease. So because of the difference in bioavailability, the dosage is also different. The main thing that is our goal for this is that how the bioavailability can be increased. Because if the bioavailability is increased, then the dosage can be reduced. So here basically what is shown is how the bioavailability of the drug is affected by certain interactions. So in the y-axis, you have the concentration of the drug, and in the x-axis, you have the number of hours. Suppose the drug felodipine is taken with water, you can see that the concentration of the drug is highest, which is around 11 nanomolar per liter after 1 hour. And as time goes, the drug is getting metabolized. So the bioavailability decreases. And after around 8 hours, the drug is completely metabolized from the system. But if you take the drug felodipine along with grapefruit juice, due to certain interactions, which we won't go into the details right now, because of that, the bioavailability greatly increases. You can see that where in case of water, maximum the concentration of drug was 10 to 11 nanomole per liter. When the drug is taken with felodipine, the concentration of the drug goes up to 25 nanomolar per liter. Not only that, the bioavailability is also greater. Even after 8 hours, there is sufficient drug in the system. That is, the rate of metabolism is greatly reduced when you're taking the drug with grapefruit juice. The effect of this can be seen is that when you are, so felodipine was used to treat high blood pressure. You can see that when you are taking the drug with grapefruit juice, both the diastolic and systolic blood pressure are the drug with the grapefruit juice is more efficient. You can see the lower line is the one with the grapefruit juice. Here also the lower line is the one with the grapefruit juice. So when you're taking the drug with the grapefruit juice, the lowering of the blood pressure is more efficient. Even the heart rate, you can see, is increased with the grapefruit juice, and that helps in reducing the blood pressure. So what is meant here is that the metabolism is reduced when you're taking the drug with the grapefruit juice, and so the bioavailability of the drug increases. So how does this relate to the activity of the cytochrome protein? You can see that in healthy subjects, so what is shown here is you have, again, the concentration of drug is in the y-axis, and the number of hours is in the x-axis. In healthy subjects, this is how the drug concentration should vary. It should go up to 6 nanomolar per liter after 2-3 hours, and then around 8 hours it reduces a lot. But there is a disease which causes increased cytochrome P activity. So if the cytochrome P activity is increased, that means the drug metabolism is much faster. If the drug metabolism is much faster, you can see the maximum concentration of drug only goes like up to 1 nanomolar per liter, which in healthy patients, the drug was bioavailable up to 6 nanomolar per liter. In patients who have this disease with increased cytochrome P activity, so increased cytochrome activity means increased metabolism of the drug, which means the bioavailability of the drug decreases. So not only does the maximum bioavailability of the drug decrease, but the bioavailability becomes zero much faster. In healthy patients, the drug was available in the bloodstream up to 10-11 hours. But in diseased patients, the bioavailability becomes zero after just 6 hours. So somehow, if you can inhibit the activity of this protein, then the bioavailability can be increased, because then the metabolism will be reduced. So now that you have studied this, then you can we have identified the protein cytochrome P as a target, and then we can design, so we have done the step of identifying the target. Then we can design certain ligands and go through the SAR by NMR process in order to find ligands which will bind to this protein and inhibit its activity. And if the activity of the protein is inhibited, then the drug bioavailability will continue to be high, even in patients with the disease. So NMR spectroscopy is very important in the drug metabolism pathway. Basically, firstly, it's a non-invasive technique for studying drug metabolism. NMR technique does not harm the patient in any way, because it is just passing a low magnetic field through the patient. It is not like X-ray crystallography or so on, where you are passing very high energy X-ray through the subject. Then it has been used for the study of drug, as we have already discussed. NMR can be used both in vitro, that is in the biofluids, tissue extracts, and so on, and in vivo, that is using animal models and human subjects. So NMR spectroscopy is unique that it permits both kind of studies. So in vitro, the main fluids that are used are plasma, serum, urine, saliva, or any other secretion of the body. These are the general diagnostic fluids. If we want to study a specific location of the body, we can also use cerebrospinal fluid in order to study. So suppose we are studying the cerebrospinal fluid, and the metabolic products that are observed are different to what is expected in a healthy patient. That will give an idea that there is some disease in the patient. Or the bile fluid, or the pancreatic enzymes, or even the amniotic fluids, milk. So milk from cows are often, we can see that a certain batch of milk is causing some diseases. That is because maybe there is something in the milk that is not supposed to be there, because the cow is diseased. So then we can carry out NMR of that milk and compare it to the NMR of the milk of a healthy cow, and see, that will help us. This comparative study will help us understand there may be certain metabolic products in the diseased milk which is causing the diseases in the humans. So study of any of these specialized fluids, as well as the normal fluids commonly used, are basically the in vitro studies that will help us get an idea of the drug metabolism. So in this case, uh, many of the isotopes can be used. The most common ones are the ones we will discuss now. So proton or 1H NMR is commonly used because it has a very high NMR sensitivity. The natural abundance of 1H is 99.98%. So because it is so abundant, suppose you have only one proton in the sample, still because of its abundance, it is highly sensitive and you will be able to detect that proton. But there are certain drawbacks also. The chemical shift range of proton NMR is very small. We know it goes from only 0 to around 12 to 13 ppm. So the chemical shift range is small. And secondly, there is a lot of multiplicity due to J coupling. So we know proton NMR, there can be triplet, there can be a lot of splitting like this can happen. So because of this, what happens? This J coupling splitting that we have, the spectra gets very crowded, and that can cause many peaks to merge, and so the observation and quantification of the drug metabolites will become difficult, both due to this kind of JJ coupling and also due to the small chemical shift range. Another point is that when we are taking the 1H NMR of these fluids like plasma, serum, urine, if we are taking the 1H NMR, then there is a lot of, we know that blood consists of more than 90% water. So from water also you will get a 1H NMR signal. So then water suppression techniques like pre-saturation or gradient-based freeze drying will have to be used to eliminate the signals from the water so that we can focus on the signals from the metabolites. So that is another disadvantage of using 1H NMR. That is for in vitro. That is, what about in vivo? For in vivo, 1H is a poor nucleus because it is not possible to spin the samples in vivo. And 1H, the problem is that because of this J coupling and everything, the lines become very broad. So because of the restricted molecular mobility that is there in vivo and the small chemical shift range, the lines become very broad. Another problem is that there is a magnetic field inhomogeneity over the relatively large sample volume. So in vivo means you are putting the whole human or animal inside the MRI machine. Because we are putting a live sample in the machine, we have to use low magnetic field strength spectrometers. And these low magnetic field strengths are not able to maintain uniform field over the large sample volume. So the sample volume in vivo is very large. It is the whole human body or animal body. So over the large sample volume, these low magnetic strength spectrometers are not able to maintain a uniform magnetic field. That is a problem in vivo. In vivo line widths are also much broader than those obtained in vitro. We have already discussed why these line widths are broader. Because we are in vivo means we are looking at a live human or live the full animal or human. We are not, so in vitro what we do, we just take the sample in an NMR tube and spin the tube at high velocity so that everything can be homogenized, but we can't do that in vivo. So because of this restricted molecular mobility, line widths are substantially broadened. So these are all the problems in vivo, and overall 1H is not a good nucleus to study in vivo. A much better nucleus is 19F. So 19F has a lot of favorable NMR characteristic. It has a nuclear spin of half. So whatever is applicable in case of 1H is also applicable in case of 19F. It has relatively narrow lines. So this is an advantage over 1H. In 1H, we know that the lines get broadened, but 19F, the lines are much more narrow. Another advantage is that 19F has 100% natural abundance. It does not have any other isotope. So it has high sensitivity due to this large natural abundance. Next, another advantage over, so this natural abundance and high sensitivity are things the proton also have. But a good advantage of 19F that is better than the proton is that the chemical shift range is large. So that helps in better separation of the peaks, and the longitudinal relaxation time is much shorter than the 1H. So we know that when we are recording 2D NMR experiments, we are varying T1. So if we can take short T1, then the time scale of the whole experiment is lesser. So that is one advantage of 19F NMR. Because normally when we take 2D 1H NMR, we have to take times of 12 hours, 24 hours to do the whole experiment. But that is not possible in vivo. You cannot scan a live human for 12 hours. They cannot be put in the MRI machine for that long. So the short longitudinal relaxation time makes the time scale of the experiment less, and so it is good to be used in vivo. And because of this time scale of experiment that is reducing, we can take a lot of, we can scan the sample multiple times in order to improve the signal noise ratio. Another advantage is that many of the drugs that are currently in use have fluorine. So 90, suppose the drug itself does not have fluorine, then the 19F NMR does not have any applicability, because we want to study the metabolism of the drug. But the advantage is that many of the drugs commonly in use, shown here, contain fluorine. And so we are able to study the drug and how it is metabolizing in the body using 19F NMR. And another advantage over 1H is that most of the drug molecules contain only one fluorine, but they have multiple 1H. So studying the 1H NMR spectra is much more difficult, because so many 1H are there that the signal gets crowded. But because you have only one, or in some cases three fluorine, or you can see the number of fluorine is generally much lesser than the number of 1H. In these molecules, it might appear higher, but the molecule does not end here. There is a large molecule here which is containing many more 1H, but the molecule is having only 6 fluorine atoms. So looking at the fluorine's 19F spectra is much easier than looking at the 1H spectra. Then you have carbon 13, which the advantage is that the chemical shift range is large. So because the chemical shift range is large, the peaks will be better resolved. But the problem is that the 13C is not abundant at all. If you look at the abundance, 13C only has a natural abundance of 1.11%. And you cannot label a drug for practicality purpose. The drug cannot be isotopically labeled. So you have to do the NMR study with that 1.1 percent abundance that is there of 13C. So because the abundance is so low, the sensitivity is also low. You can improve the sensitivity to a certain extent by using polarization transfer experiments. So basically you saturate the 1H NMR signal and then transfer the polarization to 13C, which will improve the sensitivity to a certain extent, but that is also limited. And the use of 13C in vitro or in vivo NMR studies is very limited. So as we have already said that due to practical reasons, the drugs you are ingesting cannot have 13C. Suppose you are just studying protein-protein interactions, you can radiolabel the protein with 13C. But if you're doing in vivo studies, ingesting 13C is not good for the human body. That it causes many problems in the body. So you cannot have drugs which are labeled with 13C. So the practical use for 13C in vitro or in vivo is very limited. In vitro is also limited because whatever drug you have taken, that is the one which is dispersed in your blood or plasma, and then you take a sample of the blood and plasma to study the drug metabolites. But the initial drug itself cannot have 13C because that causes health problems. And so you have to deal with the natural abundance of 13C, which is 1.1%. So it is very difficult to study in vitro or in vivo NMR by 13C. Comparatively, a much better nucleus is 31. 31P has a natural abundance of 100%. That is, it has no other isotope. So it is a very sensitive nucleus compared to 13C. Another advantage is that endogenous phosphates and derivatives of phosphates like phosphomonoester or phosphodiesters, this is a disadvantage, may interfere with signals from phosphorylated drugs and their metabolites. So the advantage of 31P is that it has a high natural abundance, so it's very sensitive. But the disadvantage is that we suppose a drug has 31p, but we know that DNA, RNA, all kinds of nucleic acids, they also have 31p because of phosphodiester bonds. And so there can be some overlap of signals between the phosphorus in the drug and the phosphorus in your nucleic acids. And like we know a technique for water suppression, but so far we don't know a technique for specific suppression of the 31p in nucleic acids. And on top of that, phosphorus containing drugs are rare. So fluorine containing drugs are common. So 19F is a good technique, but phosphorus containing drugs are fairly rare. So 31p is not used that often. So overall, among all the nuclei, we see that 19F is the one that is best for in vitro and in vivo study of drug metabolism. So just give me 2 minutes. Now, normally the methods that were traditionally used for drug metabolite analysis were things like HPLC, gas chromatography, mass spectroscopy, and so on. The good thing was that they required a minimum volume of sample compared to NMR. But the problem was that these methods can be perturbed by the column. So in HPLC, you have a column through which you are passing your analyte. And definitely, if you vary the column, then the retention time and everything will vary. Similarly, if you vary the solvent, it will vary. But in NMR, the chemical shift is constant irrespective of what spectrometer you use. So that is one disadvantage. Another disadvantage is that these methods require separation followed by identification. So first you have to purify the compound, and so you will get, you can see in mass spectroscopy, you will get many different peaks. So if you couple mass spectroscopy and HPLC, HPLC will help you separate out each peak, and then using mass spectroscopy, you can find the mass of each peak and identify the peak. So two or more methods have to be coupled, one for separation and purification and one for identification. Next, you need to optimize the separation condition each time. So if you have a certain drug that you are studying the metabolism, then suppose you are taking a certain column and changing the polarity of the solvent to optimize the separation. But the moment you take a second drug, all the conditions will again change. So for each drug, you have to separately optimize the conditions at which the metabolites are separated. Often you may need multiple separations. So in one HPLC, you may not be able to isolate. If so, see, you can see here the peaks are merging. So first time you're doing the HPLC, you might get all these peaks you are not able to separate. So you have to do the HPLC again to separate these. Another problem is that these methods are very slow. And finally, they are manually intensive. So you have to constantly sit at the HPLC machine, and you have to have skill in operating the machine and so on. So these were all the disadvantage with the traditional methods of drug metabolite analysis. On the other hand, there are several advantages of NMR-based drug metabolism study. One is that it's a high throughput method. You can very quickly screen through multiple possible drugs. And so getting a 1H NMR is only a matter of a few seconds. So because it's such a fast technique, you can screen many different ligands or drugs with the protein, or find out all the metabolism products very fast. So suppose you have 10 drugs and then you allow the drugs to be metabolism, and you have to undergo metabolism, and you find each drug how they are metabolizing. So you just have to take 10 NMRs, and each NMR takes a few seconds. So it's a high throughput method. Then you have high resolution. NMR, we already know, gives up to atomic level separation of the different peaks. Then there is least effort on sample preparation. So imagine you have plasma, you're taking in vitro NMR of the plasma. So you just take 300 microliter of the plasma and 300 microliter of a buffer. And that is enough to have the sample ready for NMR. Then we know NMR is quantitative. So by taking the integral of the peaks, we can find out how many hydrogens are there and so on. NMR is also non-destructive. We have already discussed this. Only a low level magnetic field is used, which will not destroy the sample, which is why in vivo analysis can also be done. All metabolites are detected simultaneously in a single analysis. So suppose a drug is breaking up into 5 different metabolites. If you just take the plasma after the drug metabolism cycle and you take the NMR of the plasma, you will be able to see all 5 metabolites at the same time. And NMR gives you structural information also. Finally, there is little to no spectral interference from endogenous molecules in certain, in certain NMR like 19F, there are no molecules naturally occurring in the body which have 19F. So you're not going to have interference from any signal of 19F naturally occurring in the body. The only 19F will be that belonging to the drug. Then suppose 31P, you do have interference from DNA, RNA, and so on. But suppose you are taking it from a section of you're taking it from the cytoplasm where there is no nucleic acid. Then you will have no interference of 31P also. So that is basically the advantages of using a NMR-based drug metabolism study. Now, in NMR spectrometer, we know that 500 to 600 megahertz spectrometer is enough. And for doing 1D experiments, we know that just time of few seconds are taken. Then even for experiments where you have to do more advanced experiments, as long as it is 1D experiment, which is enough to find out the different metabolites, the time is very less. And you can, temperatures of normal temperatures are enough of 298 Kelvin. If you are doing 1H NMR, you do need to do water suppression in order to suppress the 1H signals from water or proteins or lipids that are already there in the body. But otherwise, the entire setup is much simpler compared to the traditional techniques. You can see here the 1H NMR spectroscopy of human urine is shown, and there are different peaks corresponding to different metabolites that are there. We will look into it more detail right now. So there is a drug called flurbiprofen, which is an anti-inflammatory drug. And in this case, the NMR is taken from a sample of human urine. So 20 mg flurbiprofen was orally ingested. And then after orally ingesting, after allowing the drug to go through the body, a sample of the human urine was taken. And it was seen at a 600 megahertz 1H NMR spectrum. You can see here that different peaks are there, and and they can be assigned to different species of flurbiprofen. So you can see that there is a species that is shown here that is corresponding to a certain metabolite of flurbiprofen that is called glucuronic acid conjugate. So like this, just from the simple 1H NMR spectra, you can find out that once the flurbiprofen drug goes through the human body, what are the metabolites that are formed. Similarly, if you, here there is a drug where you are orally administering ethoxyphenacetin. That is this molecule you're orally administering. In this case, the 13C is radio labeled, and then it is observed that after going through the body, what are the metabolites formed? So the upper uh, uh, NMR that is there is that in a healthy patient, and the lower NMR is that in a patient with acute hepatitis. So if a healthy patient is taking phenacetin, then you will see certain peaks. But if a patient with acute hepatitis is taking phenacetin, you will see that some additional peaks appear, like the 66.9 ppm. Then the intensity of the existing peaks also decrease. You can see that 58.4 is much higher in a healthy patient, but in a hepatitis patient, it is much lower. So basically, by comparing the 1H NMR of the drug metabolism in a healthy patient and in a patient with acute hepatitis, you can find out that what is the symptoms caused by the disease. And so just by looking at the urine, if you have a urine sample of a patient, how do you prove that the patient has hepatitis? So basically, the urine sample of the patient will be compared with this reference urine sample, which is of a healthy patient. And if they see that there are certain additional peaks, or the peak intensities of the existing peaks have changed, that will help in diagnosing that the patient has acute hepatitis. So why that is all about in vitro studies, basically studying the urine or plasma of the patient. But uh, sometimes in vivo studies are also required. And why is that? Basically, if you measure the drug levels in plasma or urine, it does not affect the drug concentration at the receptor site. So suppose a person, as we said, has hepatitis. So there is something wrong in their liver. But you are studying the urine of the person and you are seeing what is the concentration of the drug and metabolites in the urine. But we are not able to see what is the condition in the liver where the problem is, that is where the drug is actually acting. So in vitro studies only give you an indirect idea of the drug concentration. Secondly, in vivo basic continuation of the first point is that a method that allows one to measure the concentration of drug and their metabolites in situ, means at the position where the drug is acting, is extremely useful. So the advantages of in vivo is that you can measure the quantitatively the amount of drug and its metabolites in the target organ, because you are studying the organ that is actually the target and not the urine or plasma where there is an indirect measurement. Another thing is that you can repetitively use it on the same patient. In vitro, you take out the blood, you study it, and it's done. The sample is wasted. But in vivo, you can repeatedly scan the same patient. And in vivo, obviously, it is non-invasive because you're only applying a low magnetic field, which does not affect the organism. The drawback is that the sensitivity of in vivo is much lower than in vitro. Another thing is that only molecules with unrestricted mobility are visible. So suppose you are studying certain drug molecule, it will only be visible in vivo if the molecule is allowed to freely move. Prolonged data acquisition may be required. So a 1H NMR experiment in vitro will take only few seconds. But in vivo, it can take up to 1 hour. Like MRI, you often have to be inside the machine for 1 hour or so. Because a much lower field strength has to be used in order to not harm the living organism. So the data acquisition time is increased. Then the observed signals may have various anatomical origins. So suppose you are seeing 1H NMR spectra in vivo. The 1H NMR can come from the drug, but it can also come from the epithelial cells that line different organisms, or from other parts of the body, which are normal parts of the body, and they will interfere with the 1H NMR signal of the drug. Another thing is that there is poor resolution. So because the lines are broadened and there are many signals from, you can see lines are broadened and there are many signals from different anatomical origins, the resolution becomes poor. So if there are chemically similar compounds, it will be very difficult to resolve and they will come in the same place. So although in vivo studies have their advantages, there are also certain disadvantages. So we will look at the application of NMR in drug metabolite studies. So as we have already seen, 19F NMR is a very good nucleus to use for in vitro or in vivo studies. 19F is already present in many drugs. The distribution of these drugs, some of which are anesthetic in the brain, and pharmacokinetics of their elimination are still a subject of controversy. That is, we have not yet found this out properly. So this remains an area of research that is still open. The feasibility of in vivo 19F NMR studies of halothane in humans has been demonstrated. So studying the 19F metabolism of halothane in vivo has been demonstrated in humans. That is, it is possible. Here an example is shown of fluoxetine, which is a widely used antidepressant drug. So you can see the right side is the in vitro spectra of fluoxetine and norfluoxetine, which is a kind of byproduct formed from fluoxetine. And the left side is the in vivo peak. So you can clearly see in vivo, the peaks are much broader and they are also like less sensitive. They are merging more with the baseline compared to in vitro. And also slightly the peaks are shifted because the environment is different, because you are studying this while it is still inside the organism, and this you are studying probably from some urine or blood or other bodily fluid. So that is the difference between in vitro and in vivo spectra. Now we will look at a study of fluorinated drugs with 5-fluorouracil, also known as FU. So 19F NMR, we have already seen the advantages. Some other advantages are mentioned. Firstly, the 19F NMR is present in a lot of drugs and high doses are administered to patients. So the concentration in the body will be high and you will be able to see clear resolution in the spectrum. The fluorine atom remains intact during biotransformation. So the drug is breaking into many different metabolites, but most of the metabolites have the fluorine atom. That is, it is not released as fluoride or anything because it remains in the metabolites. 19F signals are therefore displayed over a large spectral width with no peak overlap. We have already discussed this, that 19F has a large spectral width. So that helps in better resolution of the spectra, and there is no peak overlap because 19F does not naturally occur in the human body. It is only present in the drug and its metabolites. So here you can see we have taken the drug FU, 5-fluorouracil, and it is studied in vivo. Now you can see when this in vivo NMR is taken, you are getting signals from different metabolites of the drugs, which are written here as some aldehyde derivative of the 5-fluorouracil, some acetyl derivative shown as FACET. So these are signals of different metabolites of the 5-fluorouracil drug. So you can take basically, so this is the in vivo NMR. The FU is injected. This is the peak of FU. It goes in the body and gets metabolized into different byproducts. The products that are formed in this metabolic pathway can be detected by NMR. So you can see, we don't need to go into the chemical process, but you can see that different peaks in the NMR have been assigned to different byproducts of the FU drug. Now, suppose you are taking this NMR after 1 hour of taking the drug, then after 2 hours of taking the drug. So if you take the NMR with respect to time, then the spectra will change and you will be able to uh, have an idea of the metabolic pathway. That is, at what time, what metabolite is forming or what byproduct is forming from the drug. So in vivo 19F NMR has been used to monitor this FU metabolism in the liver. So basically, patients having colorectal cancer, they were 19F NMR spectra of their liver was taken of patients having this colorectal cancer who were treated with 5-fluorouracil at a dose of 300 milligram per meter square per day. So the patients were treated with this drug 5-FU, and then their NMR was taken. So FU was getting converted. So suppose this is the NMR after 1 hour of injection of the drug, where you are still seeing some of the drug and you are seeing peaks of other catabolites. And after 5 to 6 hours of taking the drug, the peak of the drug is almost finished. It is completely merging with the baseline. And you have peaks of other byproducts of the metabolism. So this is what happens when you treat a colorectal patient with this drug and you take the 19F NMR of the patient and see how the drug is acting with time. Now, a question can be asked that if you add this compound interferon alpha, how does it affect the drug activity? You can see that when interferon alpha is taken, then the drug activity, like the right side is basically after taking the drug, you can see interferon alpha is interfering with the activity of the drug, because when it was taken, only the drug was taken, after 5-6 hours, the drug peak has completely disappeared. You only have peak of the metabolic products. But if you add interferon alpha, even after 5-6 hours, the peak of the drug is strong. That means the drug is not being allowed to be metabolized. So interferon alpha is interfering with the activity and metabolism of the 5-FU drug by not allowing it to be metabolized. So we already know about MRI, which uses magnetic resonance spectroscopy. So MRI and MRS are slightly different. We'll look into that. So basically, suppose this is a rat brain. In the absence of a magnetic field, all the spins are randomly oriented. When you put the rat inside the MRI machine, a low field strength magnetic field is passed through the rat, and all the magnetic fields are getting aligned. Then you will be able to take, so we know that when magnetic fields are getting aligned, and different energy levels form. You can see a lower energy level and upper energy level. And then at a resonance frequency, the nuclei will be promoted from lower energy level to upper energy level, and that will give you peaks in the spectra. So this is basically how the in vivo NMR is done. So in vivo NMR like this, even of the human brain, then the human is placed into the MRI scanner. You can get this kind of NMR spectra, and then you can resolve the NMR spectra to find out what are the metabolites that are there. So MRS is basically the 1D NMR spectra. You can see we don't need to look into the pulse sequence, but in case of MRS, a 1D Fourier transform is done in order to get the what we normally know as the 1H NMR spectra, that is called the MRS spectra. In this you can you will so if you do this after a certain time of taking a drug, you can study and deconvolute the 1D NMR spectra and assign the peaks to different metabolites. MRI, in this, a 2D or 3D Fourier transform is done, and you can basically get an image of the cross section of a certain organ. In this case, it's the brain. You can see you have an image of a cross section of the brain. you can get it from different directions, a cross section from the top or from the side. Basically, MRS is closer to the 1D, 1H NMR spectra that you normally know. MRI uses more advanced techniques of 2D or 3D Fourier transforming in order to give you a 2D or 3D picture of different cross sections of the brain. So you can see here MRI has been taken of the human liver. A transverse cross section is shown. The corresponding 31P MRS is shown. So there are different peaks in the MRS, which is basically, you can imagine it as a 31P NMR, and each peak can be assigned to different metabolic products. Now, suppose this 31P spectra on the here, so these are two MRS spectra. The lower one is the baseline that is there in a normal rat. The upper one is a spectra in a rat after a seizure. You can see in the upper spectra, these peaks that are NTP, these are basically like energy currencies. We know ATP is the energy currency in humans, where ATP is hydrolyzed to ADP and so on, and energy is released. In rat brain, the energy currency is NTP, and NTP can basically be hydrolyzed in order to give energy. So after a seizure, we can see that the peaks of the energy currencies alpha, beta, and gamma NTP are all becoming higher compared to baseline. If the peaks are higher, that means the brain is becoming very hyperactive after the seizure. So the peaks of these energy currencies will help you understand that suppose the seizure has already happened and is over, but the peaks will still remain in the brain. So that will give you an idea that some time ago the seizure has happened. So this kind of comparing with a healthy rat brain and seeing the difference in intensities of different peaks, or sometimes new peaks can also appear, will help in diagnosis of certain diseases. Here you can see 1H MRS spectra is taken from the cortical region at a preclinical stage in a brain having Alzheimer's disease. In Alzheimer's disease, basically, there is this protein called A-beta PP that is aggregating. So in a normal brain 1H MRS, you would have certain peaks, but in a patient having Alzheimer's, the intensity of these peaks might change. So you can see the glutamic acid peak is reducing. The NAA peak is also reducing. Instead of the creatinine peak, another new peak is forming. So comparing the MRS spectra of a healthy patient and a patient having Alzheimer's, it will give an idea as to how the disease is progressing with the metabolic state of the brain. So when the metabolism and metabolic products in the brain is altered, the disease is slowly progressing. So that will help in mapping the progress of a disease. So that basically covers all the lectures of week 10. So we have looked into the different stages of drug discovery, what are the various procedures used, and we have looked at in vitro and in vivo NMR, how they help us look into the different metabolic products after a drug is taken, and that will help in designing a drug where you can ensure that the bioavailability of the drug is high. You can also use MRI or MRAs in order to diagnose a disease, because the in case of MRAs, as you can see, the metabolic state is very different in different organs in a diseased person compared to a healthy person, and MRI will give you a much better idea of the 2D or 3D cross sections of the organ itself. So basically that is what we have looked at in this set of lectures. Now I'll go through some practice problems from week 9, which is the assignment of the previous week. One problem is that the magnitude of biomolecular association is defined on the basis of binding affinity as strong, intermediate, and weak interaction. So what are the KD values of strong, intermediate, and weak interaction? The correct answer is A. In strong interaction, there is nanomolar Kd value. In intermediate interaction, there is micromolar Kd value. And in weak interaction, there is millimolar Kd value. Another thing we saw is that when there is strong interaction, the exchange will be slow, because strong interaction means that it is very, imagine there are two peaks, one of the protein and one of the protein ligand complex. In strong interaction, once the ligand is binding to the protein, it does not want to go back to the free protein. So the two peaks will be very distinct. And even in 2D NMR, you can see there is a cross peak, very distinct for the protein and very distinct for the protein ligand complex. Now, if you have intermediate interaction, there will be intermediate exchange. That is, the two peaks are now slowly starting to merge because the interaction is not that strong. Even in 2D NMR, the peaks will start shifting. So from the peak of the protein, it will slowly start shifting into the peak of the protein ligand complex. They are no longer two distinct peaks. And if you have a weak interaction, the exchange will be very fast. You can no longer distinguish between what, so you will basically see an average peak. You will see something like this, because you can no longer distinguish what is the peak of protein, what is the peak of protein ligand complex. They are exchanging too fast. And even in 2D NMR, you will see kind of a blurred merged value. So that is all about interactions of different strengths. Then you have a question: biophysical techniques that are capable of determining the binding affinity of a biomolecular association are what? So we know that X-ray scattering will give you an idea of the structure. It will not tell you about the binding affinity. Similar dynamic light scattering, what happens is that when light is scattering from a free protein and light is scattering from a protein ligand complex, the angle of scattering is different. So you will know that whether it's a free protein or it's a protein ligand complex based on the size. But it will not give you an idea about the binding affinity. It will only tell you whether that binding is happening or not. So the correct answer is B, that is isothermal titration calorimetry. So briefly going in the technique, we know that in isothermal titration calorimetry, or ITC, there are two cells. One is a reference cell and one is the sample cell. In the sample cell, you have protein and ligand is being injected from the top. So when ligand is being injected, protein ligand interactions are happening and heat is either being released or being absorbed. So the temperature of the sample cell is tending to change compared to the reference cell. That is why it is connected to a circuit. A thermocouple is there, so that whenever the temperature tends to fall, the thermocouple provides energy to maintain the same temperature. Or if the temperature tends to rise, the thermocouple takes away energy so that same temperature is maintained. Basically, there is a change in heat transfer to the sample cell so that constant temperature is maintained as in the reference cell. So different injections are done. So different amounts of the ligands are injected, and accordingly you get different changes in the heat. So when the ligand injected is less, there is a different heat change. When you have injected a lot of ligand, there is a different heat change. So like that, you can plot dq dt with time of injection, and you will get a certain plot. You can basically take the outer side of this plot to get a sigmoidal plot. And from this sigmoidal plot, you will be able to get the enthalpy of the protein ligand interaction, the entropy change, the Gibbs free energy change. So all the thermodynamic parameters of the protein ligand interaction. So you will be able to determine the binding affinity as well. So the correct answer is B, that is ITC. Then you have a question: which of the following statements is true for complementary parameters that make a protein ligand interaction suitable for determination through NMR? So we know we saw in this class also that the NMR observables of the ligand and protein are completely different, and the protein and ligand protein complex are similar because they have similar sizes. So because a ligand has small size, the relaxation of ligand will be slow and ligand will have sharp lines. But the protein has, because of its large size, the relaxation will be fast and it will have broad lines. So a protein has, so that is there. A protein has fast relaxation and a ligand has slow relaxation. So that is correct. What about the other things? Now, because a ligand is small, it can easily diffuse. So the diffusion is fast. Similarly, because of the large size of protein, the diffusion is slow. So point B is wrong. Then if you look at NOE signs, ligand has positive NOE, protein has negative NOE. This is again because of the size of the molecule. So again, this is wrong. So the correct answer is option C. That is, a protein will have fast relaxation because of its large size, and a ligand has slow relaxation because of its small size. And the result of this is that the ligand will have sharp lines in NMR, the protein will have broad lines in NMR. Then you have a question. Which of the following NMR methods can be used for determination of protein ligand binding? So we know that TOXI is just a general 2D NMR method which is used to identify a spin system. It has nothing to do with binding. NOSI, basically NOSI all tells you the distance between residues which are close together in space. So NOSI will again tell you about the structure of the of a molecule, but it will not tell you about the binding. So the correct answer is B, that is water-logged Z. We will briefly look into what is water-logged Z. So water-logged Z stands for water ligand observed via gradient spectroscopy. So you basically have a protein shown here and a ligand that is binding to the receptor site of the protein. The protein is surrounded by many water molecules. Firstly, the water is irradiated. Then the irradiation is transferred from the water to the protein. You can see the arrows. And from the protein, it is transferred to the ligand. Then the magnetization is measured from the ligand that is bound and the ligand that is free. And that gives you an idea of the binding affinity of the protein to the ligand. Mainly, this method is used when you have a binding affinity in the micromolar to millimolar range. So intermediate to weak binding affinity. And this method relies on this transfer of magnetization between the water to the protein to the ligand. And basically, this transfer will only happen if the ligand is bound to the protein. If the ligand is free, this kind of transfer will not happen because the distance from protein will be very high. So comparing the free ligand and the bound ligand and the intensity of the NOE in both, it helps you determine the binding affinities in the intermediate to weak range. So water-logged Z is indeed a technique which can be used for determination of protein ligand binding. Then you have a question. Which of the following statement is false for STT-based determination of protein ligand binding? So for STT-based or saturation transfer difference NMR, it can measure KD of the range of micromolar. You can see that point A is false. Point A is saying the binding affinity should be in nanomolar range, whereas STT NMR is used for micromolar binding, which is intermediate range. So it is asking which statement is false, that is answer A. What about the other statements? Statement B is saying the molecular weight of the protein is supposed to be higher than the ligand. That is true. Saturation transfer can only be used when the molecular weight of protein is sufficiently different from the ligand. You can see size difference between ligand and protein should be around 15 times. So if the molecular weight of the ligand is 10 Daltons, then the protein should be 150 Daltons. Similarly, there is no signal overlap between ligand and the protein. Because in saturation transfer, we are first saturating the signal of a protein. That means we want to selectively saturate that protein signal. So if there is ligand protein signal overlap, the irradiation will saturate both the protein and the ligand. That is not what we want. So this is correct, because the molecular weight of the protein is supposed to be higher and we don't want signal overlap. Another thing is STD can be coupled with other 2D methods like TOXI or TROSI. Here an example is shown of STD coupled with TOXI. Basically, it's the same thing. One is without the ligand, one is with the ligand, and then you can take a different spectra in order to find out that what is the ligand that is binding. So indeed, STD can be coupled with 2D NMR. Finally, the residues on the ligands which are in association can be determined. You can see an example here. When you take the spectra without irradiation and with irradiation and take the difference, suppose these 3 hydrogens shown here are binding to the protein. So you will only see peaks of those 3 hydrogens in the STD NMR. So indeed, the residues which are binding or which are in association can be determined using STD-NM. So B, C, and D are correct. So the only false statement is A. So when the question is asking which statement is false, we can say A. Then the question is saying which of the following NMR parameters is not useful for determination of protein-peptide or protein-protein interactions. So now we know that NMR basically has 3 parameters which are very important for determining protein-protein or protein-ligand interactions. One is the chemical shift, which is characteristic of the chemical environment of a particular nuclei. So if the ligand is free or it is bound to the protein, the chemical shift will vary. The second thing, variance is chemical shift is one factor. Second factor is intensity of the peak. So the peak intensity is representative of the relative concentration and so the population of the nuclei in that particular state. So suppose you have 2 peaks, one from the free ligand and one from the ligand protein complex, they will have different intensities. So variance in peak intensity is a factor that is useful for determining binding. Finally, the line width of the NMR. We know that if the relaxation time is very large, then the line width becomes broad. So proteins, we know, have large relaxation times because of their size, so they will have broad lines. But ligands have small relaxation times because of their size, so they will have sharp lines. So the T2 correlation time is also important. So A, B, and C are all important NMR parameters that help you determine the extent of binding or interaction of protein-protein or protein-ligand. So the only one which is not useful is D, that is variance in molecular weight. So the correct answer is D. Then you have a question. For assessment of protein receptor interaction through saturation transfer difference will involve what? So here we are talking about the question that is asking is that the determination of saturation transfer double difference. So STDD consists of what? We will look at the STDD experiment. So first we have the STD spectra of cell plus ligand, which here is referred to as X. We will call this as X. Then you have the STD spectra of only the cell, which here it is called as Z. So here we'll call it as Z. Finally, the STDD spectra is X minus Z. So the correct answer is X minus Z, that is B. This is basically used for proteins which are membrane bound. So you're not able to purify the protein from the cell. In that case, normal STD NMR can't be used because in normal STD NMR, you need a spectra of the pure protein. Since you cannot do that, you do STDD NMR, where you have a STD spectra of the cell plus the ligand. Then you have the STD spectra of only the cell. And the difference of this, we are calling saturation transfer double difference. That is giving you the signal of the ligand that is binding. So the correct answer is B. Then you have a question. In water log C, which one of the following statements will hold true for a successful protein ligand interaction? Now, water log Z, you can remember that whenever you have a molecule that binds small molecules that do not bind with protein will give positive signals. The protein will give negative signal. And if you have a ligand that is binding in the protein, they will also display negative signal. So if you look at C and D, both are water log Z spectra. The positive signals, basically, they are not binding. But the negative signal, they are the ligands that are binding. So the correct answer is B. Ligand molecule will give negative signal when they are binding. So the correct answer is B. Then there is a question asking, in a reaction coordinate plot spanning energy of a protein in bound and unbound state, which of the following statement is true? So we will look at the plot that they are talking about. You have a protein in an unbound state and you have a ligand. In solution, these molecules will undergo tumbling and diffusion to come close to each other. And when they are close to each other, they will form an encounter complex, which has slightly lower energy than the unbound form. The encounter complex is something where the protein, so suppose you have a protein and you have a ligand which is close to the binding site, but it has not completely settled inside the binding site. So the energy is lower, but it is not as low as the protein bound complex. Once the encounter complex is formed, the ligand will orient itself in such a way that it will optimize the binding interaction with the protein. So it will perfectly sit into the binding site. Then the energy will decrease further and you will have the bound protein ligand complex. So the energy of the protein in bound state is clearly lower than that of the protein in the unbound state. That is your option B. So option B is the correct answer. Then you have a question. A protein moving from bound conformation, from unbound conformation to bound conformation may have multiple intermediate conformations, which prepares the protein to attain structural conformations, making it susceptible for binding. Each of these conformations can be defined as a major or minor population of the protein. Which one of these NMR methods present the most suitable way for discriminating across these varying populations? So we know, suppose you have a protein in an unbound state here, slowly, you have a protein that is free. There is no ligand here. When there is a ligand, it is adopting a lower energy state here. And suppose, this is the energy when the ligand has not been added. And the protein is undergoing different molecular conformations. And finally, when the protein is free, it is in an energy minimum. When it is bound to a ligand, the energy is slightly increasing. There is exchange between the free protein and the ligand bound protein, exchange between these two states. So you're calling it the two-state exchange. Now, this is a special case where the ligand bound state is lowly populated. Normally, if you look at what we have seen before, the ligand bound state has lower energy. So this is the case when in protein ligand complex, this is the general case. So protein plus ligand going towards protein ligand. It is equilibrium is lying towards this side. This is the general case. But in this case, the equilibrium is lying in the opposite side. So it is lying so the free protein has lower energy, and so the ligand bound state has very low population. So suppose the ligand bound state only has 5% population. In order to detect this population as well as the population in any other intermediate state, the best experiment is CPMG, relaxation dispersion experiment. Why this is the best experiment is because using this experiment, you can find out the structure and the population of states which have very low population, even up to only 0.5% population. So if the question is which NMR method is the most suitable way for discriminating against varying population, the method is CPMG, that is C, because there are two methods, CPMG and ZZ magnetization transfer. These two methods are two special NMR techniques which can tell you about the structure, the exchange rate, and the population of states which are very low populated states. Even if you have a state which is only 0.5% population, they can be detected by these two experiments, CPMG and ZZ. In these options, you only have CPMG. So the correct answer is C, that is CPMG. So yeah, that basically covers this. And that's all the questions I have from the assignment 9. As always, I'll upload both this video and the slides, and you can go through them. And we'll meet next Monday for the 11th live session on this course. I'll stop the video now.